move on to our next speaker, um, Erick Matsen from Fred Hutch. So he will talk about how he uses AI in his research. And as a bit of context, Erick has given multiple tutorials about agentic coding within Fred Hutch already. So today he's going to talk more about his thoughts about biostatistical research in this context. Thank you.
Hello, everyone. In my mind, AI is coming to our scientific disciplines. And I think of there being two ways in which AI can come. One is the not so fun way, and one is the very fun way. So on the left, we have the not very fun way, in which AI absorbs all of the data in the world and it generates a bunch of predictions. Maybe they're highly accurate predictions. And we try our hardest to squeeze out some post hoc interpretation of what we could have learned from these data. I'm more optimistic. I think that we are going to have a world of many small models. Each one of these models is interpretable and extends things that people have already known in the literature, provides direct predictions and interpretation. So my talk is going to be really split in two parts. Jessica asked me to talk, like she said, about generally about how AI we can bring AI in. But before that, I'm going to give a very short vignette about what I mean by a small model. And I'll give you a hint: it only has 1 million parameters. So I have the pleasure of working on antibodies. These are astoundingly amazing molecules that can bind just about anything to a high degree of sensitivity and specificity. And antibodies come from this incredible process of evolution that happens within us. So here is a picture of an antibody that is bound on the outside of what's called a B cell. And when it is bound on a B cell, it is called a B cell receptor. This red circle is supposed to schematize an antigen. So this B cell floats around your body and waits for its soulmate, which is this antigen. And when it meets that soulmate, something amazing happens. It starts dividing at an incredibly high rate, and with a mutation rate that's almost 1 millionfold higher than one typically sees in a somatic mutation. Some of these mutations, such as this yellow mutation, will make the binding better, and some of them will make it significantly worse. And after this mutation process, there's this selection step. So those B cells that are able to bind antigen are able to divide more, and those that are not die off. So what's neat is that with high throughput sequencing, we can sequence the result of all of these high throughput evolutionary stories. And so if we take the ensemble of these B cell receptor sequences that are from a single individual, that's called a B cell repertoire. And if we take the union of all of these different repertoires and look at them in public databases, there's over 1 billion of these sequences out there. That's a lot. And so it's not too much of a jump to think, well, maybe we can take this big pool of sequence data and learn something else about antibodies, which is antibodies as protein molecules that have things like stability and binding. So the community certainly thinks that we can do this. And so they have developed lots of these so-called antibody language models. So this is sort of like ChatGPT, but for antibodies. And these have something like 100 million or 1 billion parameters and are trained on something like 100 million or 1 billion sequences. And they are uniformly trained with an objective like this. So this is the math language model objective. So imagine we have all of the sequences in our big database that's sitting out there, and we grab one sequence. We mask a location in that sequence at random. So this used to be a Q, but we mask it out, and we put it in the math language model, and we get out a prediction. So this is basically trying to, and then the model is trying to learn what that antibody site was. So here it's trying to predict the Q, and it's scored. And it's nice. Now I'm a biostatistics symposium. This is trying to do some sort of maximum likelihood estimation. It is trying to maximize the likelihood of generating the correct thing. So it's actually doing some sort of density estimation. So how might it succeed at this task? So one thing it has to do is it has to memorize the components that are used to make these B cell receptor sequences. These are germline encoded. It should learn the codon table, which, if you know about the codon table, has to do with how the nucleotide sequence translates to the amino acid sequence. It should learn something about the neutral mutation rates, so something about how mutable each different site is. And then after all of that, it should learn something about functional properties. What makes an actually good antibody? But there's all this other stuff that goes on. And that's funny because if you look at how these models are, are benchmarked, they're benchmarked in terms of functional properties. So I'm giving a very short vignette, and I'm just going to describe how these models are learning the per site neutral mutation rates. And again, you don't need a biostatistician to see that there's some correlation between these two things. So here there's the neutral probability of the non-synonymous mutation. So this is just the pure mutation rate. On this axis is the probability as assigned by one of these large language models. So we can see these are highly correlated. And again, that's interesting because these antibody language models are benchmarked in their ability to predict functional things in how the antibody performs in the lab. So the prototype of one of these benchmarks is to predict a table like this. So this is called a deep mutational scan. So the way these things work is you take your protein of interest, and then you change every single amino acid to every other possible amino acid, and you see how does that change the thing that you're assaying. So in this case, it's expression. So these blue tiles, they say if you change this site to this amino acid, then it's going to be good for expression. These red tiles say that's going to be bad for expression. So the task that's being used for this benchmarking is to basically predict this table. So this is how one of these language models performs, and not very well. There are some things it gets right. So here we have sort of like this seems a little bit lighter up here and down here. Maybe there's something going on over here, but overall it does not look so good. So now I'm going to show you a scatter plot that's going to allow us to dissect why exactly it's not doing very well. And the scatter plot is going to be composed of each one of these cells, except on the x-axis is going to be the measurement, and then on the y-axis is going to be the corresponding prediction from the model. So this scatter does not look especially great. And the reason it does not look so great is because it conflates these mutation processes and the selection processes. So here we can see if we color by the mutation rate, we can see that basically what could have been a nice correlation is getting pulled apart by things by this mutation rate. So for instance, this one is judged as being probable under the model just because it has a low mutation rate. And this one down here is judged as being improbable because it has a low mutation rate. So again, it's what's going on here is that we have an evolutionary story and it is composed of a mutation and a selection process. And the mass language models are trying to predict the combination of these two processes, even though what they actually want to predict is just these functional properties. So what are we going to do? Well, we're biostatisticians. We're going to come up with a probabilistic model. And so we can think of these mutation and processes, and we can just assign terms to them. So that we can have a P, which is going to be our mutation model, which is a small convolutional neural network. And we can have a selection model, which is a transformer, like these other language models, but it's parameterized in such a way that it's just doing the selection component. So this is where I sort of say that I know that not everybody here, there's a biostatistician, the biostatistics audience, not everybody does this sort of probabilistic model, but this is the sort of a prototype of the type of model that we all know and love. There's some classical model, and this is indeed a very old style model, but I'm giving it a 2026 upgrade by just using the tools that we have available to us using AI. So what are we actually trying to train? We are trying to predict this child nucleotide sequence given the parent nucleotide sequence. Because of that, we don't have to memorize the germline genes. We use the knowledge of the codon table. You can look this up on Wikipedia. And then we're going to use our knowledge of the per site mutation rates, and that allows the model to only learn the effects of selection. And this right here in the middle is the result of our model, and you can see that it's actually learning some things about how these tiles are laid out. This is again the ground truth, the expression. This is our model, and this is the mass language model. And I just emphasize that everyone is doing this mass language modeling thing because it's just what you do. But I think, these community, our community here, these are the people who think about how we can set up models to reflect biological reality. And then we can just bring in all of these new tools in order to enhance our research and generalize things. So now I don't have to have a regression model that's parameterized in terms of a vector. I can have a regression model that's in terms of an image or a sequence or whatever it is that I care about. So that is really just a little vignette about what I mean by a small model. So my model is small in the sense that it's just a million parameters as compared to these 100 million parameter models, but it's still isolating a very specific thing. So now we're going to step out into the void. This is a part of the talk I've never given before, and we'll just see how it goes. So now I'm going to talk about how can we integrate AI into generating these types of small models. So, the first thing, and I don't think it requires any introduction, is that there is this thing called agentic coding. And I hope that people are aware of this. I have given, as Jessica said, talks about this. There are programs like Claude Code, and this is a wonderful thing. And it's a wonderful thing for individual contributors. But I'm a PI. So, how do I use agents as a PI? So, we're going to have a little show of hands here. Who here is a PI who has directed people who have generated some sort of code at some point? So maybe directed staff members or, come on, Jeff, you can put up your hand. Staff members or students or whoever. They have generated some sort of code. Chris, you've never, okay. Okay, Ricky for sure. Okay, so keep your hands up, keep your hands up. So the people who have examined every single line of the code that has come out of your lab, you can bring your hands down. Wait, you examined every single line of the code? Okay. Okay. So I have a name for all of you, and it's going to sound like a pejorative name, but it's not. You are vibe coders. So you can take your hands down. So vibe coders are people who ask for things of the code and they don't examine the code. And so all y'all are vibe coders where the agents are the people in your lab. And that's fine. That is fine. But I just want to say, like, this sets us up for thinking about, like, how can we use agents like a PI? Jeff wants to take a picture of that. Yeah, yeah. Okay. Um, so I have been thinking about this a lot. Really, I mean, essentially since the models got very good in basically like December of last year. And so my solution is this, and I will walk through this. This is sort of a complicated slide, but I just want to start by saying that there are two things that that PIs typically do. They discuss and come up with ideas for things, and then they review the results of those experiments and things that have been done. So those are highlighted in yellow. The other thing is they know things about the literature, and so this has typically been a weak point for agents and LLMs because I think many people have had this experience where the model says something like, "I know that paper super well," and then it makes up a whole bunch of nonsense. So that's not what we want. So yeah, so I work a tool for this, and it involves this beautiful tool written by the AI people at Allen Institute that's called ASTA. Uh, so we're just gonna do a little demo here. Uh, so let's see. We're definitely gonna allow that. So use the BIP lit skill and ASTA to grab some snippets. But I'm embarrassed because of you. Uh, imputation. So anyway, we're gonna let that cook for a little while. This is like a cooking show. And we will go back. Okay, so I'm gonna walk through this a little bit. So so this is something I've never talked about in public before. But so this is a system that allows us to use lots of agents in order to agentically code like a PI. So so when I think about how I like to work with humans or computer agents. I like to think that they're sort of like the the manuscript is sort of the unit of the idea. And then there's sort of all the experiments that are happening. And so these get divided into what's happening on my laptop and what's happening on a remote server. So first, there's a skill that basically initiates a session in which the agent learns all the things that it reads the manuscript that I'm working on. It looks at all the issues that are corresponding to it, and it brings this all into context so that it's a suitable discussion partner for planning what's going to happen next. So that creates an issue. So in GitHub terminology, an issue is just a unit of work. It could be a bug fix, it could be an experiment, it could be a new code feature. That, all gets checked by once you come up with an idea for an issue, this unit of work, it gets checked by another agent. That gets submitted to GitHub, and then that moves over here. And then so there's another agent over here that's sitting and supervising all the different work that's happening. Um, and then that, agent can spawn off to some workers that are working on different things, and then those things get spawned off. There's like another sort of issue lead agent that keeps track of where things are. And then when there's a surprising result, then there's another agent that comes in that called the surprising conclusion skeptic that then investigates things and tries to understand what's going on. And then at the end, there's another agent that does a PR review, also comparing it against the Constitution and other things like that. So um, the point here is not to talk about this specific like setup. It's to talk about some principles, and the principles are that one that I have found that many agents are much smarter than one. So this. Oh wait, actually, okay. So I'll say a little bit about how this is set up. It's a big collection of Claude Code skills, which are basically descriptions of how Claude Code is supposed to do a thing. And then there's a command line interface that does things like connects with the Semantic Scholar thing, which is this Allen Institute thing, connects with GitHub and Slack and remote servers. Why don't we just check and see how this is going? Okay. So it says, okay. So here it has found two key papers. There's SC impute and add impute, and it has grabbed some quotes from those. What, Jessica, these look like reasonable quotes? Okay. So it's just to say that, like, I think we get angry with agents when they don't know everything, but if we give them tools so that they can learn things, then we can go about our business. So this again is sort of a way in which the agents can become reasonable discussion partners. I'm expecting to see something that says zoom up here. Okay. Um, right. So the lesson number one is that many agents are better than one. So even if the agents are saying something quite absurd, another agent will be able to come by and call it on that. Um, and the lesson two is that agents need to be able to work. So I have found it really wonderful to be able to create a space that the agents can work quite independently. And that actually looks like this. This is a desktop that I have that the agents can trash. My 5-year-old took this picture and me hugging this desktop because it's a great space for things to be able to work. And so, as we think about what we need to be able to do at Fred Hutch, I hope that we can create some sort of safe space for agents to be able to work semi-independently, and we can all become PIs. So that is where I'm going to end. Again, I hope the 2 points land. That on one hand, AI doesn't mean we need to start doing something totally different. It means that we should have superpowers to do the things that we were doing previously in a much better and more general way, and that AI is there to assist us in doing these things. Thank you. Yeah, Allie.
So, gonna ask a similar question that I've asked before. I'm gonna sound like a broken record, but maybe because I'm representing UW Biostat, that's not too bad of a thing. Because I'm gonna ask you about evaluation again. So I could ask ChatGPT or Claude or whatever, instead of having my own set of agents, I could ask it to also pull up snippets from Jessica's work, and sort of maybe it's different sort of from what the system that you've set up would produce. how do I, again, sort of how do I evaluate? How do I sort of decide which one's better? What are the criteria that I'm using to decide sort of which direction to go?
Let's see. So there was a lot of components in my talk. So you're not talking about validating the antibody model. You're saying like if we have a technique that allows us to look at papers, how do we validate that it's good?
I'm just talking about that as an example, but sort of more generally, sort of, I mean, even the antibody model. I mean, so you showed like result that shows that your models are doing much better in terms of recovery with much smaller sort of parameters. But sort of more generally, you could imagine that sort of foundation models could do similar to a task that sort of you set up with like smaller models with like um, carefully designed based on biology. At the end of the day, sort of what is the direction that we go? I mean, so how do we decide whether sort of we should invest more on sort of using these large language models and fine tuning them potentially in settings that we have, or sort of building our own sort of sets of small models that sort of we basically agents that we put together or sort of models that we put together? I guess that's my general question.
I will try to give you a general answer. Well, let's see. I mean, again, I mean, with respect to like the big models versus small models, I mean, I think like a correctly specified small model will always outperform like the very, very big model. I mean, when it's encoding something that is real about the biology. And I also think that that's sort of the unit of knowledge. The unit of knowledge is that model. I mean, you have brought biological insight using your work, and so that is some unit of knowledge, and that's sort of what we should aspire to. That is understanding. I mean, as far as I mean, for the like the tool that allows you to grab things from the PDF, that's I mean, I think you try it out and like does it do what it is that you want to do? And I don't think these are sort of one shot things. The nice thing is that you get to have a dialogue and ask things. That was just one question, but you get to ask additional questions. Yeah, I think Bill has.
So, how do you think of the tension between doing great training great scientists? And in particular, we're at a transition time, and pretty soon these agents will be better than any of your graduate students. So then you can really do better science if you just spend all your time mentoring your agents and not mentoring your students.
Yeah,
Oh, you should answer first. I was gonna say, I have an answer, but wait.
well, I mean, I think that can we be honest that like it's always been a bit of a mixed value proposition of bringing people into your lab. Like I love writing my own code, and oftentimes I can write my own code faster than a trainee can, and I love working with trainees because I love mentoring. And I think that that is gonna have to become more of a thing. I think, it's gonna be something that we just have to value as its own independent contribution.
Can I add something to this? So I've been thinking about this so much right now, recently, just the last month or so, Eric. I think we we have to learn what you're doing. All the PIs have to be PIs because we have to train people, our students, to be that. And we can't, so no matter what, we have to we it's not going to be about training them to be what we were. It's about training them to be what we have to figure out to be. Is that are you Yes.
I totally agree, and I feel like, getting over the hurdle is what I see for most people the biggest. There's 10% picking up, and what is the 90%?
Well, but I mean, I think maybe to flip Bill's question on its head, like, maybe it's time for us to start listening to our trainees. Our trainees are probably quite good at these things, and so maybe, we can bring. Exactly. So it's like we each bring something to the puzzle. I mean, I have loved computer programming since I was just a tiny, tiny kid and have been obsessed with, the like, command line and so on. And so I know not everybody is that way, but there's sort of a meeting of minds, right?
Yeah, I'm off topic from that topic.
Great.
If that's still going. First of all, this is an amazing talk, and the second part totally worked. And so, just I know this is your first time, but I hope you give it lots of other times. One of the questions I had for you is I've experienced the same thing, which is well constructed, handcrafted, artisanal small models beat out just dumping all the data in, and like there's certain modeling assumptions and knowledge that an expert can bring to improve make a big model smaller. My question to you is two parts. One is, you can imagine how that same kind of thinking could shrink the size of large language models themselves potentially, which would have lots of consequences for costs and all that kind of stuff. Have you given any thought to like how that might play out for like the really, really big models potentially could also be shrunk? And then the second question is, to what extent can you have the agents take away, like you've already automated your grad students, now can you automate you in the sense that can you get it to tell you like here's the abstraction you should be thinking about?
Let's see. So the first, I'm afraid I don't have anything to say about like, is it possible to have a refined, smaller version of a model of like one of these really large language models? I suspect it is. I mean, I think that we are probably like everything is going into the big models. I mean, like they add like chess data to GPT. They add all sorts of crazy data, and then that with the idea that it's all building in this sort of super intelligence. I think we're going to see some speciation where you are going to have these specialized things that are for one use or another, but it doesn't make sense now because uh like everything, the big model is getting smarter at everything so fast. Um let's see. The second question was about
You broke it up into like selection, mutation and selection, and you knew to do that to build smaller models for each of those components. Could the model have told you in advance this is the right way to break that up and make
not
smaller?
yet. Not yet. I mean, I feel like I push the model pretty hard. I mean, like in some of these, like there's like an issue next skill, which is sort of like, what should we do next? And you know, sometimes it has a good idea, but usually the ideas are pretty incremental. It's sort of like, well, we could try this, but it doesn't. Um, I mean, that said, like like once it is truly transformative to have the agent be able to access the literature. And I love to show this ridiculous interview in which the original Travis something or other, the guy from Uber, talking about vibe physics, and he is like chatting with GPT late at night, like, and he thinks he's come to the boundary of physics, and it's just complete nonsense. And so I always think about that when I'm brainstorming with one of these models, that they want to sort of tell you that what you've had is a really great idea. But once they actually they have contact with the literature, I think they do become quite interesting brainstorming partners. I mean, I wouldn't say I have any projects right now that are entirely in that direction, but definitely assisted by that.
So thank you, Eric, for the wonderful talk. I just want to conclude with maybe one last question. Since your title says thinking like a biostatistician, I wonder whether the process of decomposing kind of a black box model into many small models can be thought of as I'm just brainstorming, like a variance decomposition problem in statistics. So yeah.
Well, I'm thinking very broadly. So yes, I mean it is a factorization, but I think that there's all sorts of clever factorizations that biostatisticians have used. So
Yeah.
when I say thinking like a biostatistician, I'm thinking about leveraging all of that existing knowledge that people have and supercharging it with like the tools that allow them to do regression over arbitrary spaces.
Yeah, sounds like Bayesian.
Yeah.
Yeah. All right, thank you. It's